The next one is about outbreeding depression, and I think Linda Broadhurst is going to lead that, and I think Liz has also got something.
So just going to talk mostly about inbreeding and less about outbreeding. And I think, as Ari says, sex is good. It mixes up diversity. It mixes up genetic diversity is one of the reasons why it's good. It kind of doesn't create new genes. It creates new gene combinations. New genes come about through a bit of a different process, like through mutation. But it does create new gene combinations because many organisms inherit a set of genes from their mother and a set of genes from their father, and depending on those two sets of genes, what they inherit can be quite different to what their parents have got. So for plants, there's a variety of inbreeding systems, and they range from selfing to outcrossing, and a lot of plants actually do both. They're not shy at a little bit of selfing. And then some plants are clonal, which is where Liz is going to come in when she talks about her clonal plants. The way a plant reproduces, or its major influence during its pollination and seed set time, influences both the quality and the quantity of the seed that is produced. And so I was just going to give you an example, a species you probably know down here, the button wrinklewort. This is some work done by Andrew Young many years ago now, but it still serves as quite a good model species for us to keep working on. So it's one of those, if you don't know it, it's a small understory grassland species. It's relatively moderately lived. It's had this divergence between the two populations, up around the ACT in New South Wales and Victorian for quite some time, we think, but we're not really sure. The real issue here is, though, that small populations, and a small population for the button wrinklewort is 200 plants. So for some people, they're thinking that's already a big population. But we know in populations of less than 200 plants, we're getting low seed set, poor recruitment, and the populations are ongoing decline. And it's basically because of this reproductive mechanism they've got called self-incompatibility, and it's run by a couple of different genes, not very many. And so in a large population, this is a mechanism that's designed to stop selfing. So if you think of this as a big population, anything that's orange is a similar and not compatible genotype with the big flower at the bottom there. So we're thinking that's an orange genotype. So that means that it can't self-pollinate and it can't mate with any other orange flower in that population. But because there's lots of other different colours, which mean lots of different genotypes, this plant can actually set lots of different seed, which is great. The problem in a small population, though, is that you often find small populations are highly related. Seed often falls very close to the mother plant, not much seed actually. Even in wind-dispersed species, the majority of seed is still relatively close to the mother because often seed hits something and falls. So we get long-distance dispersal, but the majority of seed often falls around a mother plant. So in a small population, you've got this very highly concentrated number of orange plants, which means that you actually get a lot less seed produced because there's just not that diversity in the population. So that's one mechanism. The opposite end of the scale is something like in the peas, and this is the example of Swainsona erecta, very similar attributes to the button wrinklewort. Here we've got populations ranging from 10 to 400, but we still get seed set, whether it's 10 or 400, seed set's still pretty good. The problem is we've still got poor recruitment and the population's still going into decline, those very small ones. And that's simply because we're producing highly inbred seed. And so the diagram on the left here, the plant on the left of the left is an inbred plant from a population of less than 10, and the plant on the right is from an outbred population of more than 200. So you can see has a dramatic effect in terms of the quality of the seedling that's being produced, whereas the previous one, it was the amount of seed that was being produced. And then some of you may know Andy Guerin from up near Golden Broken CMA. He saw this talk and he went back and tested some of his own slender bitter peas and he's found exactly the same effect. So I think it's a really nice way that you can demonstrate to yourself whether you've got an inbreeding effect in the seed that you're collecting, is to just do a simple experiment like this. He kept his collections all separate, he grew them all at the same time so that they're exactly the same experiment, and you can see that when you've got 2 plants or 10 plants, you get a very poor result. When you've got 100 or 50 plants, you get a much better result. Very simple demonstration. So just a bit of a summary. So small outcrossing populations, you get low seed set, low genetic diversity. Small selfing populations, poor seed quality, so poor seedlings, still low genetic diversity. And then I just put up a bit of a running sheet of, this is really hard to tell who does outcrossing, who does selfing, but there's some rules of thumb. So daisies are generally self-incompatible, exceptions of course, but generally self-incompatible. Eucalypts try to sit in the middle, but generally outcross. They will self if they have to. If they've got a small population, they often go to selfing. Acacias we call self-incompatible, again there's exceptions. Banksias range, as do grevilleas. Outbreeding depression. So this one's actually much harder to demonstrate. It's experimentally quite difficult to demonstrate, and so the evidence is there that it does occur, but it's actually not as strong as we'd like to think that the evidence is for it. But one of the things I put this slide up for is because it may take a couple of generations before outbreeding depression shows up. So you might bring two populations together and it all looks pretty good, except for a couple of generations down the track. And for a long-lived species, that's probably not going to be in your lifetime. So that's going to be in someone else's lifetime that there's a negative effect. And I think this really demonstrates that the things we do now must be, the data of what you're doing must have a really long-term legacy. You know, you have to start thinking about, if I'm doing something now, how will someone in 150 years know what I've done? How are they going to know where I got the seed, how I planted it, and what the effects that we saw in the first generation might have been? Oh, sorry, yeah, sorry, I should have done that, shouldn't I? So outbreeding depression is when you bring two sets of populations together that have been divergent for quite some time. So there are good examples of where you've got quite divergent populations. If you bring those together and they mate, what you can find is that the seedlings produced from that mating are actually less fit, so they might look spindly like that previous example, than either of the parents. So you actually, it's a kind of a way of dumbing down fitness, like you get less fit progeny even though the parents look okay. Does that make sense?
Is that the opposite of hybrid vigour?
It kind of is the opposite of hybrid vigour, yep. And then we also talked about this morning about plasticity. So this is a nice example. It's from the European literature, but it's an example of how plasticity works and why it can be hard to demonstrate some of the effects that we're looking for experimentally. And so each of these plants is the same genotype. So each of these colours represents the same genotype. So they're all cloned, these plants, and then they were planted at 3 different elevations. And as you can see, they all responded quite differently depending on the elevation and the genotype that was used. So it makes predicting how things are going to respond to an environmental variable, in this case it was elevation, quite challenging. Liz, over to you.
Clonal, that's it.
I just wanted to thank some of the people who've contributed to this work over the years. What I'm going to do is just raise a couple of issues around seed collections and clonality, more to think about that rather than to provide any answers to it. So I think we've already been through the fact that plants can respond to change by adapting to the conditions as they change, or they may shift their geographical range. They may do a bit of both. And the adaptation depends on having the relevant genetic diversity, and to shift range, the conditions have to be suitable and the plant actually has to be able to disperse. So most plants do reproduce sexually. There are plants that reproduce clonally, but most of those also do produce seed. Some produce as much seed as a sexually reproducing species, and then you've got the other extreme where you've got sterile species that only exist now because they can reproduce clonally. And I just wanted to make the point that where you get that overlap of clonality and seed production, if you're not aware of the clonal reproduction or you don't know the extent of clones, then even gathering seed from what appear to be different individuals may actually result in you collecting seed from the same individual. And so if that's the case, you do risk having a seed lot that's lower in diversity than you might think. And if that's being used for revegetation, then already you've reduced the genetic variation in the gene pool of what you're restoring, and that may have effects downstream on how well that population can survive, persist. So over the last, decade or so, there's been work going on looking at the genetics of some species down in the Gippsland Lakes because of the changes that have occurred. So back in the 1880s, this entrance at Lakes entrance was opened permanently, so that's meant that over the previous 100 or so years, the salinity's gradually increased and the water level has increased, and there's been less fresh water going down from the 8 or so rivers that feed into the Gippsland Lakes, and so that's changed the conditions, and there's been a lot more impact, and so the land use has changed, and that's had an effect on species, and there's been clearing and various other things, but a lot of those areas are now being restored. 3 of the species that have been part of this are Melaleuca, Phragmites and Triglocan. So Melaleuca and Phragmites both grow clonally and both set seed. Triglocan isn't clonal, but it's quite long-lived, and so it can have a long-term genetic effect. So just to, you can probably see the Melaleuca without much to it there, Phragmites on the edge and Triglocan growing in the water in this particular instance. So what we were interested to look at was the genetic diversity in Phragmites and Triglocan. It hasn't been done for Melaleuca yet, to see whether the Triglocan that's found mainly in the wetland areas was more isolated, depending on which wetland it came from, or and whether the Phragmites perhaps had less genetic structure across the lakes, because we know that the main dispersal agents in the wetlands are water, wind and water birds. So looking at Melaleuca, I think, Randall, this plant was from the Western District, is that right? Wilson's Prom. So you can see, I think this is interesting, it's a huge individual, but you can see that it's grown in 3 stages, if you like. And this picture on the left just shows how many shoots can come up from roots. And so it's a very dense clone once it's established. Phragmites, again, can form very dense clones. This picture on my right is down in the Snowy River in some fairly saline situations. The picture over here is at Sale Common, where you've got different clones flowering at different times. And the seed production can be quite unreliable in Phragmites, and we think it might be to do with some incompatibilities depending on what particular individuals are flowering together, whether they're a compatible cross. But we don't know a lot about the breeding system of Phragmites. And the other thing about Phragmites is that it's polyploid, so it's got, as far as we know, the ones in Gippsland have about 8 sets of chromosomes, so that gives them lots of options for variation that might just tweak some of their responses to different conditions. And then Triglocan can be very common in the wetlands. This is all Triglocan in the water here. It does break off and can be dispersed vegetatively, but on the whole it's a seed dispersed species, but most of the seed is found in the wetland that the plants are also found in. So we were looking to see what effect that sort of seed production would have on the genetic structure across the site. So we know that these species can change in abundance depending on the environmental conditions. And so for the Gippsland lakes, it's the hydrology, the salinity, changing in salinity, and the amount of water flow. And part of that has interrupted the opportunities for their reproduction. So I wanted to use this video of Randall's, and he put this together, and it just shows the change in abundance of melaleucas and Phragmites over a period from aerial photos. And you can see that once it gets going, no? Oh, but you didn't. Oh, you didn't. Oh, okay. All right. No, there should have been another file. Gradually, over this period of time, this area has just become almost completely melaleuca, but it's the same little dots that expand. So over this period of time, most of these seedlings that recruited before 1957 just became larger, and there was no other recruitment of any great extent. And so this just shows that changing the hydrology can change the abundance, and that also affects the seed produced, the diversity of the seed produced. And here we know from genetic testing that these are individual plants that have just grown bigger over time, and what reappears in the middle as the centre of the plant dies out is actually just reshooting from the original clone. So these are very old plants, and they can have a big effect. The other thing that we looked at was the distribution of maternal lineages. So they give you an idea of the seed dispersal. And what we found was that some were very common. So this star haplotype was maternal lineage was found very widely, and it occurs right up through Queensland and is very common. There were certain lineages that, like this triangle, that were found just in the Gippsland lakes and tended to be in the salt areas. So then we were interested to see whether that meant there were salt-tolerant lineages, because there had been a bit of a push to identify those for revegetation. But, you know, is that a good idea? So we sampled from very salty areas down in the Gippsland lakes here, and from low salt areas, and then looked at the response of genes en masse when those plants were subjected to different salinity levels when they were growing in the same common garden. And what we found was that there was a massive response of genes when you changed the salinity levels, but there was nothing that really made it simple to say, yes, these particular clones were salt tolerant and these ones weren't. And I think that just is an example of just how complex these plant responses can be to something like salinity, and also that their use in revegetation and restoration is that really you need more than just the ability to tolerate salt. There are other issues, and so we don't want to focus too much on one aspect of a plant's makeup for an area this size where you want a lot of diversity so it can be adaptable. So just from this, we didn't find that there was any reason to restrict the selection of Phragmites. There was no structure across the Gippsland lakes, which is what you would expect for something that is wind dispersed for pollen and seed. Although we did find that the closer plants were sampled together, the more similar they were genetically. And that's because the seed and the pollen are sourced more closely to each other. What we don't know about the Melaleuca is just how genetically variable it is. But because those plants stay there for a long time, they don't have to actually reproduce or recruit very often to still have a lot of genetic variation retained in those populations. And the triglocan, surprisingly, was not genetically distinct across the Gippsland lakes or significantly. And we think that probably that's because there's more dispersal by water birds than we had thought at the beginning of the project. So where we thought that there were probably rare events where flooding spread the seed, we actually think that birds have a much bigger role in dispersal. And then finally, really, we know that the habitats in the lakes that are being restored probably bear little resemblance to what was there when some of the remnant vegetation was recruited. And so, you know, the whole basis for doing the restoration really is to try and find out how best to source germplasm so that the changes that occur mimic as much as possible what would be a natural response to changing environmental conditions.
Thank you, Liz. And for Linda as well. Thank you. Now, I reckon inbreeding and outbreeding depression are really big deals from the point of view of whether you decide to grow local provenance plants or whether you try and bring in genes from elsewhere. So I'm hoping that there are lots of questions and issues arising on Vox Vote. Do we have some? It takes us a couple of seconds to fire up.
You think I'd learned by now. So through the conversations that we're having, there was a lot of things about testing and making sure that when you put your plants in and when you bring in genetics or out, that you should monitor. So I thought it would be good to ask the question about how people in the room are actually collecting that information. And the answer is that they're relying on the nurseries quite a lot. So, and they're relying on the integrity of that information that's being kept. So I think that's probably something that we need to have a conversation about, is if we are concerned about moving provenance around and we're not actually keeping good records for our restoration projects, is that a problem?
All right, that's good. Any other questions to raise with Linda or Liz or anyone else on the panel? Well, I've got a question, I think. It seems to me that when we've got clonal populations like Phragmites australis, which grows in both hemispheres, and it doesn't produce much seed. It does produce some seed. And when I've tried to collect seed, I only get a tiny fraction. But I've never thought that's a concern, because a plant will last centuries, I reckon, and it reproduces principally vegetatively, and it seems to be doing pretty well despite having, on the local scale, a low genetic diversity. So does it really matter that it's got a low genetic diversity? And I can think of a range of other species that might be in that category. So can one... Come up to the microphone. Thank you, Liz.
I didn't mean to say that the Phragmites had low genetic diversity, because I think across the Gippsland Lakes it's a highly diverse species, but in a small location, you may only have 1 or 2 or 3 clones. So the number of genetic individuals can be quite small. But because of the dispersal of pollen and seed by wind, it's connected across very wide distances, a couple of 100 kilometres, and that is similar to what has been found in European and North American studies too.
A supplementary question. So if I decide that, based on precautionary principles and so on, I'm only going to grow the few local genotypes, is that going to create a problem, or do I really have to consider bringing in genes from elsewhere for a species like Phragmites or Melaleuca ericofolia? And more generally, how do I know when I need to?
That's very tricky. I suppose we don't know about the genetic diversity of Melaleuca, so I'm not going to comment on that one just at this stage. The Phragmites tends to have a recruitment period under disturbed conditions. I think that's fairly common, and you get a lot of seeds germinating, but you only get a few clones persisting. And so I suppose it really depends on whether your precautionary principle in using just those clones that are there is going to suit a location where the environmental conditions are changing. In the Gippsland Lakes, the reason that Phragmites is a species that actually is in need of restoration in some areas is because the salinity levels have increased up the Gippsland Lakes, and so in some cases there are clones that would have established and been growing previously in reasonably freshwater conditions, but now they're being lost because they don't have the capacity to respond to the salinity.
For an arbitrary species, how do I know whether I need to worry?
I think you have to think about its breeding system, if you can even estimate that from its morphology. And I think you should talk to somebody about it.
Liz, before you disappear. So in a sense, when you've got a clonal species, of course, as you pointed out, occasionally you do get 6.
Or in a lot of them.
Yeah,
a lot of it.
And certainly in clonal species. So for instance, we've looked at Garnia radula recently, and of course in that situation we have evidence of past 6 in some populations and absolutely no evidence of 6 in other populations. So you might have a population that consists of a single genotype, and most of your genetic diversity is actually carried in a couple of populations. So in a sense, those populations that carry that diversity can become incredibly important. And there are a number of clonal species that do that. They do occasional 6 when the conditions are right. And then, of course, those populations, you want to get a handle on where they are, because they carry an awful lot of diversity. So in a sense, you could revegetate with anything, but of course those populations that carry a lot of diversity become really important from a refugia point of view.
And I suppose the other thing is why you have populations that are a single genotype. Are they ones that are at the edge of the previous range, and so they have got to the point where they can't reproduce? Or is there some genetic reason where they have become sterile, and if you put them into another population, it would affect the fecundity of that population?
Yeah, that's a good question. And the other thing, Graham, if I may, just to make one point about outbreeding depression, and I think Liz did a good job of, Linda did a good job of covering that. But the important thing to realise about outbreeding depression is that in the F2 generation, Linda showed some graphs that showed a slight decrease in fitness at the F2 generation. And of course, what also happens at the same time is that the variance increases markedly in the F2 generation. And effectively, that means that you get far more diversity in the F2 generation, which of course will be selected against. So just because you get a little bit of outbreeding depression doesn't actually mean that you're going to have a disastrous consequence for that population, because natural selection effectively will take care of that. It'll remove those unfit individuals from the population as part of a natural process. It's a little bit like the successional process at the evolutionary scale.
Thank you, Ari. But before you put the microphone down, I've got a question that relates to the Garnia radula you mentioned. Was it in the Grampians that there was only 1 or 2 genotypes?
Correct.
Angle C?
Correct, yeah.
Okay, let's think about the Grampians since the last ice age. So if there's only 1 or 2 genotypes, 1 I suspect, if there was only 1 there, wouldn't that have been there since the last ice age? Wouldn't that one single genotype have been capable of coping with huge climate change that's happened since, and all the other changes that we've, as Europeans, launched upon the vegetation? And if it's able to cope with those sorts of changes, does it matter if we don't inject any new genes? And more generally, how do we know when we should?
So I think from the point of view of Garnia, I mean, it's certainly the case that clearly we have a very fit genotype that occurs in those areas, across the Anglesey area and the Grampians area. And that's terrific, right? Absolutely terrific. It's probably got there as a consequence of, as Liz said, selection in the past favouring a range of genotypes, and probably means that that's a very fit genotype. So that's an important genotype from a revegetation point of view. But don't forget that the first question we started off with was, of course, maintaining genetic variation. And just because you've got one genotype, it doesn't mean that you can't get some plant rust that'll come along at some point in time and knock it out. Because, of course, typically from an evolutionary point of view, they have quite short lifetimes if you only have one genotype. So it still is critically important to maintain those populations that have multiple genotypes. And like I said, there are a couple. There's one in particular that I can't tell you where it is, because apparently it's a bit of a secret for those people that are into Garnia. But that is a very important population to preserve, you know, and we need that sort of information. So I'm not saying don't revegetate with your single genotype from the Grampians, because it's clearly a good one, but also make sure that from a local point of view you are conserving that diversity in that sexual population.
Thanks very much. Now, does VoxVote give us any other things that we need to deal with?
There's one very good question, which is from someone in the audience, and I'll read it. It's long, so I'll read it by word. Without lab-based testing, is it possible to determine if fragmented populations are historically genetically divergent and unsuitable to be combined, or if they have been separated by humans and can be mixed without risk of deleterious genetic consequences? One from the practitioners, I think. Okay,
where's a geneticist who can answer that? Ari again.
Please feel free to take this. Closer? Is that better? Yeah. Okay, good. So the answer is that if you actually collect enough information genetically, you can easily get an answer to that. Because basically what we can do these days, and we can do it genomically, so we can score thousands of genes quite easily these days across a population and give you an exact answer in terms of variability. So we can tell you how much gene flow is occurring between populations. We can look at the past history of those populations based on genomic markers. So it's really a situation where if you give us the DNA, we'll give you an answer.